Thank you very much, Chris. Happy to be here. I wasn't sure yesterday I'd be here in person. If you'd like to hear about the adverse effects of too much snow, I have lots of good photos about that. You'll see the topic is very broad. I'm going to try to cover the whole range of food ingredients and constituents and contaminants and how the issue of adverse effects is looked at across that whole range. So it's a pretty broad look at the topic. This is my disclosure, and you'll see the treatment of the subject is entirely my own fault. No one else to blame. All right, so these are the questions I'm going to try to pursue for you here today a bit, and I hope we continue with them. So we want to avoid and minimize adverse effects, so we have to know what they are if we're going to do that. And that's the whole objective. And so most food substances will, at some concentration, some exposure level, cause adverse effects. And some actually will cause adverse effects if exposures are too low, the nutrients to be specific. So the questions we're going to look at are approaches to identify adverse effects. What are available to identify intake levels at which adverse effects can be, we think, not eliminated, but at least controlled to an acceptable level. And then finally, what difficulties and inconsistencies arise because of the necessary differences in approach across different categories of food substance. And that's going to be a big part of my subject. The third bullet here is perhaps the main point of what I want to present to you today. To set the stage for that discussion, I have two sort of opening slides that I want you to keep in mind as we proceed through the discussion. The first is, I think, and Chris hinted at this, we've got different categories, I call them categories, of substances that can be found in food. And we need, I believe, different decision models for dealing with them, that is, dealing with them and finding ways to minimize risk, minimize the likelihood of an adverse effect for different categories of substance. And I group them. I think it's useful. Some may disagree, but I think it's useful to talk about, let me begin on the right, category C, substances which we intentionally introduce. The list is there: food and color additives, grass substances, animal drug residues. There's something important missing, I just noticed. Pesticide residues are in this category as well. I don't know what happened to that. Food packaging materials. These are intentionally introduced, and I say that they are readily avoidable in the sense that if we think they are unsafe for some reason, we can just stop using them. Now, that's an easy thing to accomplish. It may not be easy for the manufacturers who rely on those substances, looking for substitutes and so forth, but it's easy to control. In the middle category, I put substances that are not readily avoidable. Chris, did you have a question? I'm sorry, you were looking.
Just looking at it.
Okay. Substances that are not so readily avoidable, the contaminants of food. These are widespread environmental contaminants, even natural contaminants like mycotoxins. And then also the very important category of compounds formed during processing and cooking. These are materials which, if they pose risks, we cannot simply eliminate them. We have to find other ways to control them. And so it's quite different from category C. And then finally, what I call necessary and unavoidable, substances which are naturally occurring in foods and the nutrients, of course, food itself, if you like. So that's a category A substance, and these three categories I suggest and try to demonstrate require quite different approaches to decision making to find ways to minimize risk. That's one point I will make as we go along, so keep those categories in mind. And then again, to set the stage, I have a definition, a pretty widely accepted one. I gathered this from a number of different publications. It's not easy to find this clearly set out in any one publication, I must say, what an adverse effect is. But exposure to a substance begins with some kind of physiological or biochemical alteration. And then if that leads to a structural or functional alteration, if that first step leads to some structural or functional alteration that impairs health, we call it adverse. So that can be somewhat simplified. They can happen at any stage of the growth, development, and life of the organism. They may be permanent or reversible. Simply because an effect is reversible does not mean it's not adverse. Some are not. There's a judgment call there. They include detrimental effects on behavior and learning, a very tricky subject to be sure. Effects that reduce the capacity of an organism to respond to additional insult, immune system effects, for example, are adverse. Risk assessments then are applied. My fourth bullet is the third big bullet, to data concerning adverse effects to find what we think are safe levels of intake, or we look at risk as a function of exposure when we begin with the data on adverse effects. So we need to know what we're looking at in order to decide how to get to safety. And then finally, if we decide that certain responses do not impair health, are not adverse effects, then we can ignore them in risk assessments. That's sort of the premise. That last bullet is sort of one by difference. That is, if something is not adverse, then you find it and it does not meet the criteria for adversity, then we call it not adverse. So maybe that's not the best addition for something that's not adverse, but that's the one that's sort of in use. We get information, most people know this, on adverse effects. We are limited in our capacity to develop information on adverse effects. We cannot do anything intentional in humans to uncover adverse effects. That's just not possible. So we rely heavily on animal studies and on observational epidemiology. Those are the two principal methods for turning up adverse effects. We may learn something from clinical trials, but we don't go in them to learn about adverse effects. And then case reports, we have a lot of those around, but they're awfully difficult to interpret. You can't ignore this other information, but the main bodies of information are epidemiology studies and animal studies. They are quite different in what they tell us. This is toxicology 101, but just keep in mind that these two different evidence domains, I call them evidence domains, epidemiology, animal studies, you could add other evidence domains here as well. Our ability to do studies, what we learn from the studies, the extent to which we can investigate adverse effects differs between the two evidence domains. If you look down the list, and this is far from a complete list, there are other issues as well that arise in these two different ways of uncovering evidence. All the advantages go to animal studies, except the very last one, wrong species. But keep in mind, too, although we can study effects in humans, we often are looking at effects in limited segments of the human population. So even that is limited. So these are limited areas of investigation, yet we have to work with both domains of evidence here and try to make something of them. I'm going to begin by looking at category C because this is the most well-established area for looking at safety, avoiding the adverse effects of substances. Work on this began in the 1950s, and it has continued to this day. If we go through that very briefly, I will move to the other categories, because you will see in introducing the first category that we have not struck a good path to deal with the other categories, not a complete path, I should say, to deal with the other categories, because the way we approach category C is not going to be useful for the other categories, I will argue. Okay, so category C, the intentionally introduced substance, we rely very heavily on animal data for this purpose, to understand adverse effects. Couple of the reasons, there are a lot of reasons actually we do this. One is that under our laws, we're supposed to understand adverse effects before we introduce something into food. So the only way you can do that is through animal studies. No other way. And then we're supposed to have a pretty complete picture of the adverse effects of a substance, something you cannot undertake in human studies. So animal studies here, they have those strong advantages. There are others as well. Animal testing, then, has been the major source of information on adverse effects of things we intentionally introduce into food. There are, after 50 or 60 years, you can look at what now is assumed about adverse effects uncovered in animal studies and what we can assume we can learn from those studies. There are lots of things we assume. I call this areas of science policy. That's a term from the old National Academy Red Book from 1983 on risk assessment, where you've got some scientific basis for an assumption, but it's not complete. But you are paralyzed without the assumption. You cannot say anything about risk without the assumption. So you make the assumption. These are often called defaults. These are some key ones from animal studies that I call science policy. I introduced these because I'm going to ask the question later, if we get into other categories of food substances, is it clear that these same science policy assumptions should hold? So adverse effects in animal studies are assumed to be relevant to humans unless we have, in specific cases, data, mechanistic data or something to say they're not. So we make the assumption they're relevant. It's not even important to be able to say that the animal data predict specific types of effects in humans. They're still used for purposes of safety assessment, even if we cannot claim that a cancer animal predicts a specific cancer finding in humans, and we cannot claim that. Okay, that may seem odd to many people, but it's a common assumption. High-dose effects are taken for granted, unless, again, you've got reason to discount them. So these are all sort of default assumptions in the risk assessment process, and the only way that we get to use animal data for adverse effects is to make them. The last one is kind of interesting. Keep in mind, there's a lot of things you cannot learn from animal studies that are important health effects. And this is just a partial list. So there are limits on animal studies, but there are other assumptions as well. But keep these in mind. I'm going to ask the question, as I said, whether we want to hold on to these same science policy assumptions when we look at other categories. This is the typical dose-response analysis mostly from animal studies, where we move on the dose increasing from left to right, from no observed effects relative to controls to frank effects on the very far right. That's an old term that most people have forgot about. FELs, frank effect levels, clear toxicity. The no observed adverse effect level is where there may be some effects, but they're not considered adverse. They are adaptive or compensatory. We move then to what might be an ambiguous level of exposure where you're getting some changes and you need to make a decision about whether or not they are adverse, whether they lead to health impairment in the long run. You might label that either a low observed adverse effect level or no observed adverse effect level, depending on your judgment at that point. So that's always going to be some uncertainty about levels of exposure and whether or not they are adverse. Everyone knows this. You've seen this formula. We find the no observed effect level from animal studies, and then we worry about the fact that of variability, that humans may be more variable in response than animals. And within the human population, there's more variability. We use these, again, these are science policy judgments, these uncertainty factors of 10. They're not grounded in pure science at all. And we use them as a standard default assumption, but we then look for alternatives if we've got some scientific data to do it. But we end up with something called the ADI, an allowable intake, which we think is safe. It is not we call this risk assessment, but in fact, it's not a risk number. It is a level we think to be safe. And in fact, we do not, under current methods, try to quantify what that risk level is. I think we should, and there's work going on that says we should define ADIs as some level of acceptable quantifiable risk. And it's possible to do that whether you have a threshold or non-threshold effect. But that's not in the pantry right now. We use the ADI, and it has a long, good history of use. I'm not saying it's not, there's a problem with its use. I just think it can be improved. But that's our factor now, which we use to define safety. And if the estimated daily human intake falls below the ADI, we say the substance is safe. If it doesn't, we can act to remove it. It's a very simple model, and it's one that's been around and worked pretty well for a long time. What I call a bright line decision model, safe, not safe. And it's that simple. So now let's ask the question, what problems might arise as we attempt to move into these other categories of food substances with this model? Consider here a set of issues that arise when we do this. And to do that, we've got to look back at the what was in the minds of those who developed this simple ADI model as we begin to look at other approaches. And keep this in mind, that at least these factors were not considered in the development of that original bright line model. Number one, that some substances we may have to deal with pose benefits as well as risks. Category A substances are certainly in that category. Two, some might be under homeostatic control, and that is because of the various mechanisms within the body to control exposures. Some in these categories might be under homeostatic control. That was not in the minds of anybody who developed the original ADI model. They may decrease or increase risks of multifactorial chronic diseases. Again, category A substances, we'll hear more about that later. They may pose effects only detectable in humans. The model does not consider that. And results from high-dose animal studies, particularly for natural constituents of food, have not been very well analyzed and looked at. And in the same way that we have looked at results from non-essential chemicals. And then finally, in the last category, the substances that are contaminants of food, or these processed chemicals are just maybe not readily avoidable, they should require consideration of multiple control options, different kinds of interventions, and look at how risk is altered by those interventions. Where do we get the greatest benefit? That's a different model than just drawing a bright line. And finally, I ask this question, I don't have an answer to it. Do we really want to apply the same policy judgments we use for category C substances for category A and B substances? Does the science really say that those assumptions make sense for the other categories of food ingredients? Consider category B substances. It's easy to say, well, I can derive an ADI for aflatoxin or something like that. And is that a solution to the problem? I don't think it's much of a solution. We need better solutions when we're dealing with contaminants which are not readily avoidable. We need to look at how different ways of controlling them, different interventions, affect levels and then affect risk. How does risk change as we intervene in different ways to control contaminants? With many of our contaminants as well, we again get into other evidence domains because the important contaminants of food, the PCBs, the mycotoxins, and others that are important and regular contaminants of food have a lot of epidemiology data associated with them, not so for most additives. And so that raises a lot, and by the way, most of that epidemiology data comes from occupational studies. You can't ignore it. Acrylamide, we've got a lot of occupational epidemiology data. You just can't ignore that. So the question is, how does it fit with the other evidence? How do you integrate it with the other evidence? Those are wholly new problems and also have to do with how, what's really adverse. I will argue the bright line model is not useful for this category of ingredient. We need to look at how risk changes with different kinds of interventions. Think hard about how we can best control a problem. Look at risk under existing conditions, and then look how that then changes as we try to control the problem. Then make a decision, the criteria for decision making here, the best approach to mitigation. We answer questions like what risk reduction is achieved. We need models that look at risk reduction as a function of exposure, not just bright lines. Are there indirect effects of an intervention that might increase risk? And then what technically achievable intervention achieves the greatest benefit, also considering perhaps cost. There's an ILSI workshop last summer on process form chemicals that has this as one of its platforms. And we have a mycotoxin workshop coming up in the summer that again looks at this kind of approach to dealing with these complicated problems. Enough about that. Let me do a little bit about A. This is the greatest challenge. The natural constituents, the nutrients as well. A lot of work, as Chris mentioned, going on this. We've got here risks as well as benefits in at least the nutrients and probably for many of the natural constituents, the so-called bioactives as well. That's a different kind of model than and where we try to look at how we're going to get to minimizing risk when you're talking about both benefits and risk. That is a much more complicated problem. Again, we've got different categories of evidence used here, and evidence integration from different sources of evidence, different domains of evidence is an important topic. It's getting a lot of attention these days in other areas of environmental science. How do you integrate evidence best? And to do that, one thing you need to understand is how the adverse effect is identified and defined in these different areas of evidence. If we're going to do this and do this well for complicated substances like natural constituents of food and nutrients, we need to be able to integrate well, and we also then be able to say that we've got some consistent approach to identifying and describing adverse effects. So here's a great ILSI project, I think, where you could look at this question in some detail, look across different areas of evidence, from human evidence to animal evidence and mechanistic studies, and how within those fields of science are adverse effects identified. How do we decide whether any observed response is adverse and is not, how severe it is, how then that is looked at, whether effects on various biomarkers are adverse or not. I think you will find across these different evidence domains not a lot of consistency on these questions. So it's a great idea, I think, to look at this as a possible way to improve situation, develop some common criteria where it's possible. Sometimes it's just not going to be possible because the evidence is so different. What you can learn from animals is not always transferable to people and vice versa. But you've got to make this clear, I think, and it's not clear now, I think, in a lot of areas where we're looking at evidence from different categories and trying to make sense of it and trying to integrate it. We'll look for some kind of harmonization here may be desirable. Final slide, or the next to the last slide, on mechanistic understanding. There's a great deal of work going on here now, you know, and looking at sort of high-throughput testing of chemicals, using in vitro assays, suites of in vitro assays, low exposure testing, testing in the range of human intake, and then we try to make some assessment of it. This kind of testing and other studies of mechanisms of action have great explanatory power, and they should be used much more, I think, in, for example, the nutrition area or the area of bioactive chemicals than we have used them. Because when you explain phenomena like adverse effects, your understanding goes up a lot. And so explaining them requires mechanistic understanding. One thing this kind of study does not tell us, though, is what the adverse effect is. It may explain whether an effect observed is adverse or not by understanding the mechanism. That's a great way to explain effects, but not necessarily the way to identify effects, this high-throughput testing. So one of the issues that will come out of the high-throughput testing world is the effect seen. How does it translate to something adverse in people? Not clear at all. But other mechanistic studies tell us a lot, again, to explain adverse effects. And then finally, just a sort of a summation, just a repeat of what I said before about the category C substances. I think it's important to then begin to break down food into these different categories and think of different decision models about using evidence, identifying adverse effects, and then using that to assess risk, using different models for decision-making across the three categories. That doesn't necessarily mean inconsistency. It just means that there is a demand for a different approach when you're dealing with things which are, as I said, readily avoidable, things which you cannot avoid, must live with, and then things that are sort of avoidable to a certain degree but not completely. And I think those three categories, I hope, are useful in thinking about this problem. I will stop there. Thank you.